Welcome everyone to Inside the Lab with Phenomenex. My name is Brian Rivera. I am a product manager, and today I'm very pleased to be joined by three of my colleagues, subject matter experts, if you will, on the world of synthetic oligonucleotides. So really excited to get into it. So before we begin anything, I'd like my colleagues to introduce myself. Michael, would you like to start?
Yeah, sure. My name is Michael McGinley. I run our global technical services group here at Phenomenex. Been with the company about 18 years. My background, I'm actually a protein biochemist by training, and kind of got, for lack of a better term, about 20 years ago, I got sucked into the nucleic acid separation science field. And since then, I've managed, in Brian's position at times in the past, and managed many of our oligonucleotide separation projects, as well as several of our bio portfolios throughout the years. So nice to be along.
Great, thanks, Michael. Here.
Yeah, thank you, Brian. Myself, Mihir Thakur. I'm also the product manager for the bioseparations product line. And so myself and Brian, we both are responsible for designing the strategies and the different innovative product lines under the biologics portfolio. Before that, I used to be a part of the a lab guy. I used to handle the many monoclonal antibodies and other glycosylated protein analytical workflows and characterizations. So it is very exciting to be the part of this product management team. And thanks for having me here, Brian, and welcome all. We'll be hoping for the exciting sessions. Thank you.
Great, thanks, Mihir. And finally, Ivan.
Hey, good morning, everyone. I'm Ivan. I'm an application scientist here at Phenomenex. I'm basically the hands-on guy who's done a lot of the work in the lab. I've been running a lot of the recent oligonucleotide experiments with our comms. And my previous background, I used to work at a next-generation sequencing company, and I have a education and background in biochemistry and structural biology. So I've been trying to bring that to Phenomenex and see how we can expand the portfolio. And yeah, a lot of the hands-on stuff has been me. So if you have any questions, let me know. Thanks, Brian.
All right, great. So the way this is going to work is you all will submit questions and we will do our best between ourselves to hopefully address them. Again, we are talking about oligonucleotide separations and characterizations. And so I thought in order to center the conversation and to make sure we're all talking about the same thing, synthetic oligos, very unique, especially myself. I'm very much like Michael and actually Mihir. Protein biochemist, you think, okay, well, large molecule, I'm used to that. I'm used to protein, but oligos are almost a whole nother world. So Michael, maybe you could start off just by talking about some of the unique challenges that oligos face and the way that we go about characterizing them by LCMS.
Yeah, sure, Brian. I mean, the biggest thing is that, yes, what do they have in common? Nucleic acids versus proteins and the like. And they're all biopolymers, of course, but that's about where things end. When we think of it from a chromatographic point of view, proteins and the like are usually associated with, having some degree of hydrophobicity to them. Most of the amino acids are. And, obviously there's 20 different unique, chemical entities with proteins. When we shift to nucleic acids, we're basically talking about, 4 different nucleic acids that we're working with, whether it be DNA or an RNA. And, we're not talking about something hydrophobic. We're actually talking about something that's highly polar. Actually, from a chromatographer's point of view, it's like the nightmare molecule to be looking at. And we're talking about something that you have a nucleic acid base, which is only mildly hydrophobic, and it's basic. It's connected to a sugar, which, as we know, doesn't even retain on reverse phase chromatography. And then to make it even worse, they're linked with phosphate groups, which are, make them polyanionic. So you've got something highly charged, you've got something mildly hydrophobic, and that basically you're either going to separate by ion exchange or, reverse phase. But in reverse phase, they don't stick to reverse phase columns for the most part. Yeah, they need some help. And so generally the way that folks have found to make it work has been by using an ion pairing agent. to actually assist in increasing retention of these molecules, as well as giving the ability to separate these molecules by their length, because obviously the amount of ion pairing that binds onto the oligonucleotide increases or decreases based upon the length of the oligonucleotide present. So that's part and parcel of what gives us the power to be able to separate these molecules. And that's far, far different from reverse phase, where actually of proteins, where we're actually looking at the effects of each individual amino acid on the retention. So it's quite a bit different.
Oh, that's great that you point all those things out. From you know, we're a column vendor. Separation science is what Phenomenex does. You know, what would you say would be sort of the requirements? Because you know, if you think about a C18 is a C18, but can any standard C18 work for this type of work?
Oops, sorry, I was muted. In general, you know, what we found over the years in the industry. Well, first things first, we tend to run these things in these ion pairing mobile phases, which usually are neutral or mildly basic pH. So, you know, one of the things that has to come into mind whenever you go into this world is that, you know, you have to have columns that are suitable of dealing with mildly alkaline conditions. We tend to, because hydrogen bonding is a big factor in oligonucleotide behavior, we have to address those issues, usually using a higher temperature when we do our separations, to really try to disrupt some of this hydrogen bonding behavior to actually get some pretty decent chromatography. So as a like, if you actually look overall, some of the best performing columns for doing oligonucleotide separations, generally they've been C18s. C18s seem to be the flavor of choice when we talk about that. But they want to have characteristic that seems to be to show as some of the better performing columns seem to be some of these hybrid materials. Yes, they handled up the slightly alkaline pH a little better at high temperatures, but selectivity wise, they seem to be the ones that seem to give the best selectivity for oligonucleotides.
Oh, that's really great that you point that out. I mean, it's you almost have high temperature, you have high pH, you know, at least in the world of LC, right? Anything really above neutral is going to be high pH. And then also that difference in selectivity as well. You know, I mean, oligos very closely related impurities that are going to allude very closely to what the main peak is going to be. You know, selectivity definitely plays an issue with that. Maybe we could talk about something more interesting, something that Phenomenex has been exploring as of late with this idea of, you know, these are negatively charged compounds, the idea of chelation to certain things, recovery being an issue. Ivan, do you want to maybe speak a little bit about some of the experience that you've had, you know, with the bioinert flow path and how that might impact the separation with oligos?
Yeah, a lot of the recent work that we've been doing with the new biotitanium hardware has significantly improved the chromatography. We've sent samples out for mass spec at other facilities, and they've come back with very positive feedback saying that the bioinert hardware has considerably reduced chelation of metals. There's considerably fewer adducts present, and compared to just the bare stainless steel columns, and generally been an overall positive response.
Yeah, that's great. Yeah, it's it is a really interesting thing. I think in the world of oligo separations, being at that basic pH, since you're not pushing with that 0.1% formic acid like you typically are in any reverse phase situation, you know, any displacement of any trace metals is going to be a concern. You have that bioinert flow path. That's definitely going to help. Again, just to remind everyone, feel free to submit any questions that you might have. And so just take a look here. So actually, Michael, this would be a good question for you, just because I know I've been asked this in the past, is what's the largest oligo you have analyzed by reverse phase LC, and what are some chromatographic considerations that would come into play here?
Well, I think basically to start the answer to that question, Brian, and it's good for people to temper their expectations, is you mentioned something about how selectivity is really important in this world. It's a struggle because what you have to remember is that oligos, especially with some of these therapeutic oligos, the things we're trying to separate and purify from them are chemically very related to what we're looking at. They are just missing. You have a 21 mer, an N-1 is just missing one base. So it's 20, 21st similar in chemical identity. The challenge is that's bad enough to start with. The challenge is, let's say you start going bigger. Okay, so now let's get to a 40 mer. So in a 40 mer, now you basically, the difference between an N-1 and a 40 mer is 39 to 40. So it's 39/40, it's the same chemical identity as your full length oligo of interest. So as you get bigger, the selectivity challenges get more and more brutal. When we get and use the standard typical 100 angstrom type pore cell type materials, we really start, the resolutions get narrower and narrower until we get to about, we do okay out to about 100 mer. We seem to be able to handle 60 to 100 mer with the 100 angstrom pore type materials, and they seem to do all right. We played up in there. But now these days in the CRISPR-Cas9 world, life's getting more interesting because we guys have folks who are working with probes lengths that are up to like 200 mer in length. 15 years ago, if you said a 200 mer, everybody would laugh at you because you couldn't synthetically make an oligo that big to any felicity. When you start playing over 100, now the difference is, like we say, 99/100th the chemical identity of the main impurity from the full length. It starts getting messy, and there's a lot of N-1, N-2, N-3s that mix into it. So when we start going above 100, we can no longer really use 100 angstrom pore size material. We actually have to start using the wide pore type materials. And in particular for us, we make our wide pore core shell materials, our Aris material, the C18, the one that we've worked with the most. But the Biozen wide pore C8 could also equally be used for this. We've played out to about as far as 200 mer. And that's about the biggest one I've done. And again, anything about 100 mer, you're going to need this wide pore, wide open pores for those. At 200 mer, it's 6,000 Daltons in size. So for something to fit in, it won't fit into 100 angstrom pore size. So we played out to about 200. Are the N to N-1s baseline resolved? Heck no. There's no way they're even close. But for the most part, with some of the 200 mers I've played with and 180 mers we've played with, we've been able to see, we've actually been able to see chromatographic differences between the N-1, N-2, N-3, N-4 from the full length. So that's about as far as we get, and it's never going to be perfect. It's going to be a little bit messy. There's going to be some overlap. The concept of saying I'm going to get 100% pure, a 200 mer, I think that may be kind of an unrealistic expectation. It's about doing the best you can.
Okay, so there's a couple of things that maybe we could unpack with what you said there. So it does sound like maybe with the 100 angstrom, 100 mer really is kind of the sort of cutoff point, maybe a little bit more.
Kind of getting to the edge there. You might be able to sneak a little bit bigger, but not much bigger than that.
So, would you almost rule out anything? Obviously, now in this day and age, when we talk about mRNAs and things of that nature, these are kilobases long. Would you say then chromatography is just pretty much out of scope, or what are your thoughts on that?
Well, it's not so much chromatography is out of scope. It's like, the larger molecules get, the less intact chromatography tells you, just because it can't. It's just, it's the percentage chemical identity differences between things get when you get into very large molecules. We're talking about million or millions of Daltons in length for messenger RNA. So what can you see? Well, reverse phase kind of starts becoming kind of almost, for lack of a better term, a little bit silly. You'd be using non-porous columns and they get really big. Some folks have done some work. You could, in theory, and some folks have done this with using methacrylate-based gel filtration type materials, but the only information they're going to give you is just maybe just like, hey, it's around this molecular weight size. That's about it. CE is also kind of useful when you get down to that big, but even then, it only is going to tell you like, hey, it's in this size range. And that's just the nature of working with really, really large molecules.
Any comments on T1 RNase or digestion's almost akin to like a peptide mapping situation? Is that a viable technique to characterize some of these longer nucleic acids, not the least of which is mRNA?
Yeah, and actually that actually gets to what are people doing with mRNA. In essence, yeah, looking in the intact, maybe gel filtration, like big gel filtration columns tell you like, hey, it's about this big. Fantastic. The work that a lot of folks have been doing, in particular, I think Moderna and a few other guys have published on this, has been actually what they've had two main assays that we see them doing a lot chromatographically. Number one is they'll do, like you mentioned, Brian, like a T1 RNase digestion, where they're trying to digest it, kind of like akin to a peptide map in the protein world. And they'll do that, and they'll run that by LCMS. It works, but it's not the greatest, because you got to remember, again, we're talking about a 4-letter code, and we're also talking about some of these nucleases are not the most specific thing in the world. So
there is no trypsin in the world of nucleases.
There is no trypsin in the world of oligonucleotides. But Pat Lumbaugh, some guys at Pat Lumbaugh's group at University of Cincinnati, they've been playing a lot with modified RNases. Work looks interesting. Will it turn into something like a trypsin? Probably not, but it may make it less messy. The other thing that chromatographically folks are doing with messenger RNA is the tails and the heads and the tails of messenger RNA are really important for them to actually be processed and for them to be to engaged in generating message. The cap, they have a unique cap on the 5' end of the messenger RNA, and so that unique, what some folks have been doing with nucleus, they've been doing digestions and actually looking at the capping structures to make sure that they're correct, because that's really important for them to be work. Likewise, looking at the tail end on looking at the tail of the messenger RNA.
Right? That's all Mike Beverly's work. I think Mike Beverly at Novartis was doing a lot of that stuff.
They have done a bunch of work too. Those are the guys, the gurus there that have been playing with it. But even then, will we ever be able to get something where we can look at an intact large oligo like we do with, like we do with a small, like an siRNA or an antisense RNA? I don't know. We'll see. It's just right now. I'm doubtful, because those molecules are so big. Can you really see such subtle differences in something that big without chopping it up into pieces? We'll see.
All right. So next question we have, it's one that I think is interesting when we start talking about traditional silica materials and the temperature stability and capabilities. 60 degrees is commonly what we're talking about, but in the world of oligos, you have maybe with a duplex, a melting temperature associated with that, potential intramolecular interactions. These can be an issue. So Ivan, maybe you could talk about your experience in implementing temperature as a method optimization tool, and kind of what works and what doesn't work, and just elaborate a bit about your experience there.
Yeah, we've done quite a few runs. I've explored the full spectrum of everything from ambient to what seems to be unreasonably hot, maybe like 80 degrees on some of these oligos. It really depends on what you're working with. If you're looking at some of these GC-rich oligos that like to form hairpins, homodimers, and things like that, cranking up the temp really helps separate them out nicely. If you get a little drop in retention time as expected, but the actual chromatography improves significantly. And then when you're getting into, like you mentioned, some of the duplexes or some of the like double-stranded siRNAs and just even double-stranded like vanilla boring DNAs, increasing the temperature has really helped. And some of the latest like Biozen products, we've been running some of these columns at elevated temperatures for weeks, if not months. and they've been incredibly robust and generally showing that if you're running just about any oligo at 60 to 80, I mean, it's looking pretty good. If you start cranking it down a little bit, you can also increase a little bit of retention, and really depends on like the general sequence. If, again, GC rich stuff likes elevated temperature, some of the phosphorothiazide oligos like being at elevated temperature too, and definitely have seen a lot of improvement in the chromatography itself at the elevated temperatures, 60 plus.
So would you say that it would be a good sort of heuristic thing to adopt is to run 60 degrees or higher? I mean, you know, because part of me says, I mean, how much do you have to run if let's say it's a reasonable amount of GC content and it's not necessary to run that high of a temperature? You know, would you say you'd have to run that high?
Not necessarily. It really depends on the sequence, I think, is what it comes down to. I think there needs to be a lot of individual curating to the oligo itself that you're working with. Some of these oligos look perfectly fine at 40 degrees. Some of them look considerably improved at 60 degrees. So I think just from anyone's perspective who's running oligos just needs to take a little bit of time and run a couple of temperature gradients just to make sure that you're optimizing and catering to the sequence itself, which seems to have the greatest impact on which temperature it performs best at.
Yeah, I think that's a really important point because I think as much as it's, yeah, temperatures in general can help, but sometimes that chromatographic difference is going to be insignificant. You know, anytime that you're running past 60 degrees, you are putting in almost unnecessary strain on your column. Now, you have a oligo with a high GC content and everything else. Absolutely, you're going to want to run at that higher temp, but assessing whether or not that's something necessary is something I think worth exploring. So.
Yeah, especially when it comes to like the individual sequences. Sometimes you even just take like an antisense sequence and it doesn't behave the same way as the sense sequence. So just making sure that you take the additional time and the effort and considerations into tailoring everything to the individual sequences is important.
Great. Okay, so moving on to the next question, and I think we addressed a little bit of this previously, but can you discuss anything around chromatographic approaches to separating long, so 80 to 180 mer oligos with different hydrophobic modifications? So, Michael, I think you had sort of addressed the long mer, but I think this is a more interesting one is as we start to look at oligos with these really esoteric sort of modifications, whether that's being conjugated to a fluorophore or different phenyl or alkyl groups, a lipid conjugate, things of that nature. You and I have had discussions about this in sort of the needs and selectivity. You mentioned C18s and everything else. Maybe you can speak a little bit on, okay, if we have these different modifications, how, from a chromatography standpoint, how then do we approach those particular challenges?
Well, you know, Brian, I think that basically you have to start in thinking about like what I said the first question we came around about when we talked about dyes and labels. Is that when we talked early on, we said that basically oligonucleotides have, without the help of an ion pair, have minimal amounts of retention. They don't retain well. So one can imagine that these, you know, when you have a dye or a quencher or something like that, or a lipid on these things, if we just look at them by themselves, you know that we actually do have pretty good, from a reverse phase point of view, we have pretty good understanding of how to separate those compounds reverse phase. They are retentive. They, you know, they do have, like, different aromatic groups have different aromatic selectivity by either a C18 or a phenyl phase. So, you know, you start getting into these things where, you know, sometimes it, even though you're like, wow, I've got a 40 nucleic acids in length, and I've got this dye or whatever on it. It's actually, from a reverse phase point of view, in the absence of ion pairing agent, all the retention is going to be based solely on the quencher or the dye. Adding in ion pair into it will, like, basically you're modulating two retention characteristics: the ion pair retention of the oligo versus just the natural native retention of that aromatic molecule. Or they're usually, like you said, they're usually aromatic for dyes or quenchers. So you kind of, you know, sometimes it's better to look at your dyes and quencher first to make your chromatographic decisions, and then look at the whole oligo along with it, because you know that the impact of the sequence and the like is going to have much less of an effect on it than the dye and quencher present. And the way you would modulate between that on how, hey, I want to have more impact from the sequence and the oligonucleotide connected to these dyes and quenchers is going to be how much ion pair I put in, because then the more ion pair is going to have more retentive characteristics from the oligo present in that whole mix. So I think it's like I said, people sometimes just think of the oligo, but it's actually, sometimes I like to think you should, on those dyes and quenchers, look at the dye and quencher first in making your chromatographic decisions.
Now that makes sense. If you think about how polar, like you mentioned, how polar that irrespective of almost how long that oligo is, that if hydrophobicity is what we're focusing that separation on in reversed phase, that dye or quencher or lipid conjugated thing on the oligo is going to be the dominant chemical entity that you're leveraging to perform that separation.
And that kind of also goes into, Brian, like we've got some, when folks are, we're, especially in therapeutics, we're dealing with a morass of conjugation techniques on delivery vehicles for therapeutic oligos. We see a similar type of challenge when we're working with, like, for example, lipid conjugated therapeutic oligonucleotides and trying to do pharmacokinetics. We've published a lot about Clarity OTX and pulling out oligos from all sorts of tissues and the like. Hey, it works great on a phosphorothioate or just even an LNA or just an unmodified oligo. It works great, but all of a sudden you throw the lipid in there, and now it's like we're potentially separating the oligo from the lipids and stuff like that. But you just put a lipid on the oligonucleotide, so it's actually going to behave more like the lipid than the oligo in doing isolation in pharmacokinetics work. So it's again, these attached groups play a huge role in retentive mechanisms.
And it seems like everyone's attaching everything to an oligo, right? You have some people attaching an oligo to an antibody, which is wild. I think they call it antibody RNA conjugates, ARCs.
Yeah. So what's it going to behave like? It's going to behave like the antibody, right? Or mostly behave.
Well, actually, you'd be. So I know for ion exchange, right? I think the predominant way that you do it is anion exchange, and you would analyze them actually in negative mode. So, but we're not talking about antibodies. That's all the previous slide.
That's a different topic.
The MAB ship has sailed. We're kind of past that. So another question came, and this one is really interesting, and we could probably spend quite a bit of time on this. But so you have oligos, 21 mer, completely polar compound. Hey, why not HILIC chromatography? So here, maybe what we could do is let's center ourselves and talk a little bit about HILIC chromatography and just have an open discussion about how that might work for oligos.
Yeah, I mean, it's a really good question. So in here, this HILIC is not the true HILIC in the sense, right? So basically, what we are doing here is like we are using the power of three organic compound: acetonitrile, isopropyl alcohol, and methanol as well, right? So we consider here this HILIC is the new, not the true HILIC chromatography, but here what we are using is the power of the isopropyl alcohol and the methanol to separate the higher and the bulkier the oligonucleotides. So as Michael mentioned about the in the earlier the answers that this higher and the higher molecular weight, the longer oligonucleotides are very it's a bit tricky to separate at the terminal at the end of the chromatographic separation. So the standard oligonucleotides are very easy to separate with the IP, RP, IP, but when we are dealing with the higher or bulkier oligonucleotides, when we really need to switch that separation techniques to by adding or switching to that organic the acetonitriles or the non-polar mobile phases. So here it's really we need to incorporate that HILIC phase, right, just by switching that organic solvents. So it's not the true HILIC chromatography, but yes, we are using the power of this IPA and methanol, so that's why we incorporated this HILIC. And yes, it helps really to separate the higher molecular weight or the higher more oligonucleotides here. So it makes a significant amount of the contributions to separate the high molecular weight oligonucleotides here.
Okay, but let's say that we just have a traditional 21 mer oligo. I've seen, and to be clear, there's just been a ton of literature as of late. I know, Michael, you had mentioned before, Pat Lumbach at Cincinnati doing this type of work here. HILIC to me, it's a finicky separation. I'm not going to do it unless I have to. But Michael, do you have any thoughts about HILIC, how it applies, maybe what's the optimal phase to explore? Because I think as most folks are trying to move away from alkylamine ion pair, and it's like I got an open access system. I don't want to have to worry about this when I switch polarities on my mass spec. If you are exploring HILIC chromatography, where do we start with oligos?
Well, generally, Brian, it's kind of, in theory, if we look at HILIC, we're thinking about those exact things. In fact, I think Pat and his group went into it. I remember speaking to him 15 years ago about this exact subject. In theory, you would think HILIC would be perfect. You'd be like, oh my God, it's perfect. We don't have to do ion pair. We hate ion pair, in the protein world. We run away from it. His original observations, or the general observations, was he wasn't seeing that. The thinking was that in the absence of ion pair, that HILIC would have more higher signal, higher sensitivity. Haven't quite seen that. Seems that having the combination of the ion pair and the organic alcohol seems to really give us the best sensitivity. The types of columns and phases that we generally use is you got to think about it just from a hydrogen bonding interaction and the like. You would tend to kind of more favor, like the silica-based materials when we did this work, tend to not work well. When I'd say just pure underivatized silica. The diol type materials seem to be the ones that were when we were doing this on this experimentation, and others were doing this too, seem to be the ones that were giving the best results. The amides looked pretty good too. When we talk about the HILIC chemistries. Again, I think you nailed it. HILIC's known for being really mass spec friendly, but from a selectivity point of view, it's not like known for having the same degree of just really fine resolution that reverse phase can get you. So yeah, you can separate things.
You think about hydrogen bonding, electrostatic interactions, everything else. You can't finely tune that the way that you finely tune methylene selectivity, right?
Yeah. And like we've talked about temperature, we know temperature plays a role in reverse phase. We know that, we can play a little bit with the solvents. We play, we haven't even talked about ion pair yet, but
Oh, Michael, we're going to talk about ion pair. Don't worry about it.
There's a dozen different ion pairs.
We're going to talk all about the ion pair.
Yeah, that all have different chromatographic behaviors. So we got a lot of tools and a lot of tweaks that we can do. HILIC seems to be not quite as there yet. And that's not to say it won't be. It maybe it does, that we start getting the secret sauce of getting good selectivity out of HILIC for oligonucleotides. But it looks encouraging, but I'm not convinced yet.
Yeah, I mean, I hate to sound so averse to HILIC, but it is I think that separation itself needs to mature a little bit before we start to really have more conversations about what's the best practices for oligos. Certainly can do it, and again, the literature suggests that you can do it and even obtain sensitivity on par with what you'd see in ion pair. But since ion pair reverse phase is so established, and I think if you adjust certain mobile phase conditions, you can actually get away with the least amount of ion pair possible and still get a very meaningful separation, and still get the benefits that you would see with otherwise higher concentrations of those things. So with that said, that's a good segue into Ivan. I know you've done a ton of work on looking at two primary mobile phase components that are necessary for reverse phase separation of oligos. Hexafluoroisopropanol and perfluorinated alcohols, as well as alkylamine, the ion pair that we use. So maybe to start off, talk about a little bit about the effect of HFIP and kind of what you've seen in your experience with that.
Sure. I think initially this was approached as there have been a handful of very conventional methods that people have been using for absolute ever, with no real rhyme or reason to using just absolute expensively unnecessary amounts of HFIP. And the preliminary work was to see how low can you go before the chromatography degrades, and it turns out you can go tenfold lower than a lot of conventional methods. So conventional oligonucleotide method that I would see in the past would be 100 millimolar HFIP, if not more than that, and 16 millimolar alkylamine. And there doesn't really, the evidence that we've produced recently doesn't really suggest that you need that. A lot of the most recent experiments show that at 12.5 millimolar HFIP and even trace amounts of alkylamine, you can get perfectly good, equally good, if not better sometimes, depending on the sequence, chromatography. And I think that actually works out to a lot of people's favor in that there's a lot of cost savings associated with that. Some of the mass spec is actually improved. And on top of that, you were talking about the different alkylamine pairing. I think it's been very interesting, the work that we've done recently, in that different oligos and different oligo sequences and different oligo types behave very differently with each alkylamine pair. So just jumping into saying we need to use 12.5 millimolar HFIP with, let's say, 4 millimolar TEA, which has been an optimized method we've demonstrated, might actually look better with, say, 1 millimolar hexylamine instead, or dimethylcyclohexylamine. And so I think an important consideration for a lot of individuals who are looking to improve their oligochromatography need to be looking at different ion pair reagents and making sure that they're finding the right one for their immediate application and for the oligo that they're working with.
So what you're saying is I don't have to run 400 millimolar HFIP.
Absolutely not. That's just, if you like throwing money out the window, please continue to do so. If you don't like doing that, like the rest of us, then considerably lower amounts are perfectly amenable to very high quality chromatography.
Yeah, you touched on it a little bit. The one thing I was hesitant on with backing off on that much with the HFIP was the mass spec sensitivity. Would that take a hit? From my understanding of the way that the electrospray works for oligos, that HFIP is critical. It doesn't seem to be the case, and you can get
That's
away
right.
with, or I guess, implement 12.5 millimolar HFIP, and your mass spec sensitivity actually improves. So go figure. So I guess anything else in regards to the different types of oligos in the alkylamine ion pair that you use? Is one better than another for, let's say, a 2' MOE gapmer, or is there any other effect or any other considerations that one might implement before starting these experiments out? Because there's a lot of alkylamine out there, let's face it.
I don't have a absolute direct answer to which type of oligo, which type of backbone behaves best in which type of alkylamine ion pair, but generally, I would say, from my experiences, the HA, the stronger ion pairing reagents like hexylamine, have behaved considerably better. Well, I've gotten considerably better peak shapes than the conventional TEA. And I think what's ultimately being questioned here is like following conventions. TEA has just been the go-to. It's just what you do, right? You read a protocol, it says use TEA. And a lot of other people don't really take the opportunity to jump around. And I'd say that going to DMCHA or HA it's a quick, easy experiment, and it's considerably stronger, and you need to use considerably less of it. So just a quick.
So, yeah, and just for a little bit of a clarification there, you've gone how low with some of the ion pair here?
Oh, like half millimolar, even like 0.25. That's where it starts to break down for some of the hexylamine pairs. But even 1 millimolar HA is gorgeous.
Yeah, and so I mean, it almost makes sense in that When you back off on the HFIP, you maybe use a little bit less ion pair, as low as half a millimolar. You can still obtain good chromatographic separation. The mass spec data looks reasonably good. And you know, it is really less harsh conditions on your column, extend your column lifetime. As much as everyone wants to think that Phenomenex just wants to sell a ton of columns, and no, we want you to get the best use out of your column. And if optimizing those mobile phase conditions can do that, then absolutely. So maybe shifting gears a little bit, talking more about purification of oligos, similar in that a lot of folks implement ion pair reversed phased in a similar fashion. So the question would be, what do you think about using polymer media for purification of oligos? I've heard of people using source 30Q, which I think is source 30Q from Dow. So I think there's a couple of different things there, but so Ivan, I know you've done a little bit with purification. Maybe just speak a little bit about some of the things you've done, and Michael, I know you've done some things as well. So just more or less an open discussion of the different techniques to purify oligos, addressing this polystyrene or polymer type of media, and we can go from there.
I don't know, Michael, do you have a little bit of background first?
Michael, go for it, man. You've been doing it.
Too long. You know what? A lot of folks have been saying that. Yeah, for definitely, some of the folks seem to be, that we've heard of a couple of people out there, and we've done a little bit of work on this, is that especially for those who on the purification world, we get down, we're talking about two different types of purification. There's obviously anion exchange, source Q30, and TOSO also. There's some popular resins used for that type of work, the high surface area porous type polymer medias. That's mostly, we see a lot of folks doing that with Tritolon. For Tritolon, a lot of historical stuff is using this like 30 micron polystyrene reverse vein media, and yeah, some of the data looks pretty good on it. I've been hearing some folks even trying to do some unique ion pair Tritolon stuff. Haven't done it myself with it, but I've heard some folks saying that they really like it. For Tritolon, it's these polymer media seem to be pretty good. They seem to. I know that we've looked into this a little bit. So some of these ion pairs, some of these polystyrene medias for Tritolon seem to be pretty cool. They seem to be pretty nice. You get nice load. Then you can blast the column with acid to get the trittle off or cleave the trittle group off of the oligo, and then you just elute the oligo in moderate organic, and maybe even put a gradient in there to try to get some resolution to get your N- excess off. So yeah, it's something that we've been hearing more about lately. Obviously, we also in the trittle off world, we've got some folks. I know that some of the folks in the synthetic world who make synthetic DNAs and the like really seem to like to use more of the traditional silica C18 type materials for their type of purification in trittle off. So that's what kind of some of the stuff I've heard.
Just for clarification, Michael, when you say trittle, you are referring to
dimethoxytritol. Sorry, lazy.
Acid labile protecting group that's used for
the 5' group that's on during synthesis. That when you come off your synthesizer, you get a choice. You're like, either do I remove the 5' the dimethoxytritol group off before I clip it off the resin and collect it, or do I keep it on there and clip it off the resin? And your purification pathways will be determined based upon whether you keep it on there or not during your purification process. And everybody moved away from doing Tritolon purification probably about 20 years. Tritolon purification used to be the way people started. About 15-20 years ago, everybody moved away from it because they were saying that you saw a lot of problems with depurination, especially the DNA. But now we're starting, with RNA, depurination is not as big of an issue, and we're actually starting to see a lot of folks kind of starting to swing back and look at it again. So at the end of the day, we're a chromatography company. We're going to provide solutions for whatever people are doing. So there you go.
We even have SPE solutions that do a tritolon purification. I mean, that's
We
all the same.
released that QSP, what was it, 18 years ago or 17 years ago?
It
Exactly.
wasn't
So
that long ago. Come on now, Michael.
I'm aging myself. Maybe 16 years ago. Sorry.
Something like that.
Time
Okay.
flies.
It does, right? So moving on to another question here, and maybe we'll toss this over your way, Mihir. So, how does column dimensions and particle size work for oligos? Is there an advantage for using sub 2 micron particles or micro or nano chromatography? So maybe first just talk a little bit about uh, some of the particle morphologies and other considerations, and maybe we could delve a little bit into micro and nano because I think that's an interesting conversation as well.
Yeah, definitely. So here, the challenge is what we have discussed so far is with the heterogeneity of the oligonucleotides in a one sample. So to deal with that, the conventional, the 5 micron columns is not the solutions for that, right? So we have to go downgrade to reduce the particle size and the column dimensions to get the maximum separations for that, right? So micro and nano flow can be the answer for that, and to precisely says that this particular micro and nano flow dimensions will tackle that the heterogeneity of that oligonucleotides samples, right? So as you know that the gradient also, like folks used to use that curve gradient to separate those the mixture of oligonucleotides, but that the protonation charge with on the higher oligonucleotide sequence and the conventional 6 to 8 mer oligonucleotide sequences creates that the meshy kind of separations at the later stage of the separations. So just by incorporating the nano and micro flow to the game will definitely change the resolutions for the higher oligonucleotides sequence. So definitely, I would prefer to go with the micro and nano flow if our resources are available, and if we can implement that, then easily we can minimize the challenges what we are facing with the method development aspects. Let's say temperature, let's say mobile phase optimizations, let's say gradient, right? So many other aspects can easily be modified and just by incorporating the micro and nano flow technology. So yeah, definitely. I mean, I would really prefer if one can have the robust, reproducibles, and micro and nano flow column for that. We'll go for that, right? So we'll minimize the pain what we used to face with the other method development aspects, Brian.
Yeah, no, so I mean that's a really good point in that as we move towards micro and nano, there are a lot of benefits, a lot of considerations as well, but I think there is more of a shift towards the low flow chromatography in the world of oligos. You think about oligos and how low they're dosed, and if you are a DMPK lab doing a pharmacokinetic curve, trying to drive down to below 1 nanogram per mil sensitivity, that's difficult. One of the easier ways to get there is low flow chromatography. In addition to, you are using, from a mobile phase perspective, you're infusing a lot less ion pair, which of course resonates with everyone who's ever had to clean a source or have to deal with, switching from negative to positive mode, and all of a sudden you see a bunch of triethylamine. I know our colleagues at SciEx have done a little bit of work in the microflow world, and hopefully we can transition at a certain point into nano, although that definitely does pose another whole set of issues that we need to investigate. But so another question coming in: how can I do LCMS with oligos when chromatography typically uses a lot of ion pair reagent? I think we've more or less kind of talked about that, right? That a couple of things that we can do is optimize the mobile phase, maybe use a little bit less, maybe even microflow. Ion pair is almost fundamental and necessary with with reverse phase for oligos, and if we can use the correct ones in addition to optimizing their concentrations, the impact is a lot less significant. But I think another question to have here, I'll just open this up to anyone who feels so inclined to answer it, is this idea of passivating the system and cleaning out even glassware and things of that nature. For really one main reason is this adduct formation. Yeah, anyone that's ever seen a full scan spectrum for oligos knows that the things are really messy to begin with, right? They take on a high charge state. And when you have adducts associated to that, that can make for really, really messy spectra. So, cleaning out the system, sample considerations and everything. So, Ivan, maybe because I know you've done quite a bit of this work. You've also worked quite closely with Jason Chen at Scripps. Maybe talk a little bit about some good practices in system maintenance and passivation, and some other considerations, glassware, et cetera, that one might implement if you are doing any mass spec work for oligos.
Yeah, that was kind of a surprise to learn. Coming from like a dirty, biochem lab where you just keep reusing the same stuff for years and years, you assume that these things aren't problems, but it turns out that your glassware absorbs like tons of sodium and tons of phosphate. And simply just using a brand new bottle, or if you don't have the luxury of doing that, putting in some high purity water and sonicating it for a while, just to really get a lot of that absorbed, sodium and potassium out of there, has considerably improved or decreased the amount of adducts that we've seen, which was quite surprising. And a lot of things, just leftover stuff in the system, just a good formic acid flush of your system and your column before you're going to do some of these more high sensitivity experiments have considerably improved or reduced the adduct formation and improved the chromatography. And good formic acid wash, making sure that all of your bottles are nice and clean or brand new if you can afford it. Definitely stuff that I would have never thought would have been a consideration, but turned out to be quite major players in seeing cleaner spectra.
Any thoughts about, so I've heard sort of two different solvents being used, isopropanol, methanol being the two primary ones. I know Jason, for example, uses acetonitrile. Is there any, and maybe Michael, you can speak to this, is there any optimal sort of organic solvent that's used, presumably to solubilize any leftover ion pair that could be in the system?
Yeah, I've heard both. I've heard some folks like IPA, some folks like acetonitrile. I mean, well, the problem is some of these things are not soluble in acetonitrile. So I hear folks doing a lot of combination. THF, I've heard some folks using that as a cleaning solvent in there. You could even theoretically go crazier and get into non-miscible solvents. But like I said, IPA, acetonitrile are kind of the two that I hear the most of. I mean, have you heard anything different, Ivan? I mean, those are the ones I'm used to using.
It seems consistent.
Yeah. I mean, the big thing is you're gonna, the big trouble is that I think that especially for first timers getting into doing LC-MS and oligonucleotides, is that a lot of, and literally, I used to see this in meetings all the time. And literally you'll go in there and you always knew the ones who were just getting into oligos versus the ones who weren't. Because the ones who were just getting in, they throw their spectra up on the board and you'd see tons of sodium, potassium adducts and the like. And the ones who'd been doing it a while, of course, you didn't really see much in the way of addicts. And a lot of that, again, comes down to this whole thing of cleaning your system, understanding that if you're going to do, a lot of folks do dedicated negative ion mode systems that only see negative ion mode and oligonucleotides. That may not be practical for everybody. So for some folks, it just may be having an understanding that if I'm going to go from one place to the next place, I'm going to need to make sure that I wash my system out between, my different types of molecules I'm going to work with. And Sykes has shown a lot of similar data. I think they have two dedicated LCs sometimes. We have one LC doing negative ion mode and one LC doing positive ion modes, both piping into the same mass spec.
And from my understanding, the LC is actually the major contributor to this, right? I mean, yes. So certainly labware is one consideration, but the LC is, if there is any leftover triethylamine, it's going to be stuck somewhere there.
Yeah, and that's exactly what we've been
or leftover TFA or leftover formic acid or whatever from the for the protein work you were doing. Yeah.
Yeah, it's exactly what we've been seeing with the Scripps guys, and Jason Chen there has been absolute workhorse, and he goes between positive and negative all the time. He runs proteins all week and then turns the system over, and I think it's like only an hour and a half wash that he does, hour to hour and a half with a little formic acid and a little IPA, and the system's good to go. He flushes everything out, and we've gotten some pretty decent spectra out of him. So just making sure that you're turning things over diligently and making sure your systems are clean really does play a positive impact.
Yeah, key is just having a protocol going back and forth on what you're going to do.
That's right. Yeah, and I in general, I think that this idea of dedicating systems and mass specs might be a little bit overstated. So certainly,
yep, it's old hat.
But if I'm a LC or mass spec vendor, I absolutely want you to dedicate a system. Oh, you're starting oligos? Let me sell you a new high resolution mass spec and a new bioinert LC. Okay, so we've only got about 4 minutes here. Hopefully we can crank through a couple more questions. And again, if we don't get to we don't answer them here, hopefully we can get back to you and address them via email. But one more question here, and I actually wouldn't even know how to approach this, but maybe you all do. Any suggestions on good practices of preparing the mobile phase? Can the use of a third pump with low flow and a higher concentration of IP, so ion pairing agents, be better than preparing the mobile phase for two pump systems? So this is just a discussion of, and I know that this is an issue, preparing that mobile phase, especially solubility issues. I think, Michael, you had alluded to that before, but is there any good practices for the preparation of the mobile phase and doing any sort of weird things with the pumps? Would that help at all? Maybe, maybe not. I can fill the silence if you'd like.
I don't know. What are your thoughts? I mean, I have some very strong opinions, but what are your thoughts, Ivan? I'm before I chime in, I'm going to listen to you give your opinions first.
I don't know. I mean, make sure your agents are fresh. Make sure your bottles are clean and new. I can't really speak to that directly, other than just make sure you have quality new reagents and make sure you're doing everything diligently, checking your pHs. And I'm not really sure where this is leading to. Maybe
yeah, I mean, I think one of the other concerns is like stability of these things. I know that
make it
people
fresh.
put a nitrogen blanket over the thing. Yeah,
make exactly as much as you need for your experiments, and then get rid of it.
100%.
Yeah, yeah, I think that, I mean, like I said, I think you nailed it. A lot of these things is just, the whole, I think Ivan alluded to it earlier, glassware is really important. And that is, what happens with your, when you get done with your glassware, and oh, I'm going to clean it out with detergent. I'm going to put some soap in there. It's like, yeah, do you really want to be doing that?
No,
it's
no detergents. No.
Yeah, let's detergent shouldn't touch glassware. You should, organic and water is enough to do any cleaning that you need on these things. And you don't let the dishwashers in your laboratory take control of your glassware. Like, keep it away from them. I think other stuff, the other thing too is that, we sell them, some other guys sell them, really getting used to using those cap type things, a lot of those caps on top of mobile phase reservoirs.
Shout out security cap.
Security cap, yes, thank you. Mobile phase, these caps on mobile phase reservoirs really, first it stops carbon dioxide from getting into your mobile phase, changing the pH, but it just, it also stops particulate and dust and crap. That's just common sense stuff. Why not do it? If it potentially pulls the problem of getting bit by it, why not do it?
Absolutely, absolutely.
I mean,
we are coming to a wrap here. I did want to thank Michael, Ivan, Mihir. Thank you so much for your time and your expertise. And the world of oligos is new to a lot of folks, so feel free to reach out to us at Phenomenex. If if you'd like, you can just hop on the website and hit up the chat. You know, that's the team that Michael actually leads up, and so we'll help you out and hopefully steer you in the right direction as you're starting this this new world of oligos, if that's you, or if you've been doing it for a while, maybe we have a couple of other suggestions for mobile phase that you maybe never thought of. So once again, thank you all for your time, and enjoy the rest of the afternoon.
Thanks,
everyone.
Bye-bye.